This is Spotlight from all Things Photonics, featuring intimate sit-down conversations with preeminent figures in our industry. Unscripted, timely, and topical, this series shines the spotlight on industry-leading changemakers from all corners of the photonics landscape, from the gateways of progress to the most impactful challenges of our time.
Today's episode is brought to you by Hamamatsu Corporation, a leading manufacturer of devices for the generation and measurement of infrared, visible, UV light, and X-rays. Hamamatsu is dedicated to the advancement of photonics through extensive research and offers specialized systems for select applications. Visit hamamatsu.com to learn more.
For many years, cancer research was centered on the idea that these malignant cells operated independently of normal bodily functioning, or at least were systems that had gone awry. But Dr. Frank Winkler at Heidelberg University and Dr. Michel Monje at Stanford University have come to understand cancer as a development that depends on, and indeed hijacks, typical processes in the body and the brain for its survival and growth. Focusing on the emerging field of cancer neuroscience and the way brain cancers proceed, Winkler and Monje have embraced optical technologies to follow this progression. Winkler, a professor of experimental neuro-oncology, has used two-photon imaging to confirm that glioma cells link through microtubes, similar to typical neurons in the brain. For their groundbreaking work, the two scientists have been awarded the 2025 Brain Prize by the Lundbeck Foundation, one of Denmark's largest enterprise foundations. And in this spotlight conversation, Winkler discusses the journey they took to arrive at their conclusions, the conversation and ultimate collaboration between them, and how the shape of cancer research, particularly under the umbrella of neuroscience, is changing. Jumping right in here, because I know you and your colleagues have been working for this on quite some time. There's been a number of research papers you've been involved with in this area. But traditionally, as I understand it, cancer was thought to be an aberrant activity that operated largely independently of body functioning. But both you and Dr. Monje from Stanford University have discovered that brain cancers, and indeed other cancers as well, are highly dependent on neuronal connections for their survival and growth. Is that an accurate way to describe it?
Yeah, thank you for this session. That's actually exactly that nails it, I would say. So traditionally, we have this concept that cancer is just a very disorganized entity without growth inhibitory signals, and that's just where the tumor cells have kind of a chaotic way to proliferate, and where the brakes are kind of lifted from the normal behavior. However, we have kind of learned in the field of cancer research and oncology for quite some time that the environment of the body can have very peculiar and by and large tumor growth promoting effects on the cancer cells. So there is a bidirectional crosstalk between cancer and our normal body, and the nervous system has not been really appreciated so much, which is particularly interesting because in brain cancer, of course, the brain is full of the nervous system. That's what the brain is, full of neurons and astrocytes, glia cells, and other cell types. And so the coming from two different directions, both Michel Monje and we here in Heidelberg discovered that these brain cancers really resemble certain neurodevelopmental traits, and they show neural features interconnecting to communicating networks with each other, the cancer cell, but also with neurons of the normal brain. So for example, we discovered together that cancer cells receive direct neuronal synaptic input, and this direct neuronal synaptic input drives growth, invasion, therapy resistance of these cancers in the brain. And these with these discoveries, we now understand much better how intricate neuronal and cancer interactions can actually function and work. And now increasingly, we also understand better and better how this works in the entire body, because our body, of course, is full of nerves, different nerve types. And cancer can grow everywhere, and these specific interactions with kind of a lot of cancer type studies so far, and distinct nerve types in our body, sensory nerves, sympathetic, parasympathetic nerves, autonomic nerves. They have different actions, mostly also driving the initiation of cancer, the growth, the spread, and also therapy resistance. And all this is a fascinating field that will eventually or lead, or at least has a very high potential to lead to a new pillar in cancer therapy.
Right, a new paradigm going forward that ultimately could have significant clinical benefit if things play out here.
It's a paradigm shift. Yeah, that's correct.
Right. And of course, we kind of play in two realms here at Biophotonics. There's the research side and there's the instrumentation side.
Right.
And I understand from some of the published research that you've used a two-photon intravital microscopy system. And I've seen that you used a Tri-M Scope 2 microscope with a TI sapphire laser. Is that still the instrumentation you're using? Or?
Well, yeah. So we are now at this moment of time we're using from the shelf system. Yeah. So we are not really physicists or engineers who are really tweaking around with our own systems, but we have bought a Zeiss LSM 980, and this is with a coherent discovery NX multiphoton laser. And this, of course, the technical development in intravital two-photon microscopy helps us a lot to reduce phototoxicity, to reach deeper brain regions, to improve multicolor imaging, etc. So this is a fantastic discovery tool for us, because biology does not happen in 1 or 2 dimensions. It happens in 3 dimensions of space, and then the 4th dimension of time is super important. And that's actually how we went into this entire business, by exchanging the skull of the mouse and then glue a glass window, so you can continuously image the living brain. And that's a typical neuroscience method. So neuroscientists have used this to follow single individual neurons over months, even years. And we have used it to follow single tumor cells over hours, days, weeks, months, and even up to 1 year. And this was crucial, because by doing that, we are seeing is believing, but also seeing is understanding. So we really saw what's going on, and we saw that this tumor, by very diffusely infiltrating the brain, by colonizing the brain, and that's unfortunately what these tumors are very good of, these glioblastomas and other incurable brain tumor types. By doing that, they were extending, retracting neural-like processes, scanning the brain, and then interconnecting to a communicating network where the single tumor cells, and then by combining it with optogenetic technologies, where you can incite neuronal activity, you can nicely see how that then influences the tumor, and vice versa. So you can nicely probe and interrogate the system. You can learn a whole lot by doing that. So for us, this was really the jump start into the field, and we are using it until today. And so this is a strong focus of our lab, certainly, and combining it more and more with fluorescent reporter, so to say molecular imaging, if you like, and so really understand the 4-dimensional dynamics of this cancer organism, because it's quite similar to an organism, this cancer, right? With different cell types collaborating, talking to neurons, etc. So this is something that helped us a lot to conceptualize it, and then to interrogate it. And that's why I just want to make a strong point for photonics.
Right, yeah. Well, I'm glad you mentioned the phototoxicity aspect of it, because that's traditionally been the challenge in brain imaging, I believe, is capturing all those dimensions you're talking about, while at the same time understanding that you're dealing with a living dynamic sample, and how to observe it, and get all the information you need from a clinical standpoint. That's always been the challenge, as I understand it. And it sounds like you've made significant progress in that area.
Yeah, so that's So you're right, and that's definitely something that you couldn't do with the traditional with a confocal microscope, right? It's won't be too phototoxic, and you don't want to follow your own phototoxicity over time. You want to follow what's going on, right, in pathobiology, so to say, over time.
Yeah. I think you got into this a little bit a minute ago, and clearly your work has revealed a number of intriguing and interesting aspects of how brain cancers function. Like you were talking about, essentially the cancer hijacks the traditional workings of the brain to its own advantage. And but one of the things that I believe your research had revealed was that tumor microtubules formed, which allowed the communication between cancer cells. But I think, if I remember right from the research, that there are actually treatments that currently exist, and I don't mean to make that into more, but there's receptor antagonists that are currently on the market now that could potentially interrupt that communication and might be beneficial therapeutically. Is that accurate?
Yes, that is accurate. So yeah, we have discovered that these cancer cells extend these neural-like very long membrane tubes, in some of them millimeters in length or even longer. The human brain is really infested in these diseases by these very long membrane tubes that are very similar to axons and dendrites of the normal brain. And these cancer cells, I mean, that's how the nervous system functions, right? So you have these very long processes so that single neurons can connect over long distances, and that's how complex networks can develop and can then also function. And the tumor is doing something very similar. And yes, they are different from normal neuronal networks. They're kind of a mixture of neuronal and astrocytic networks. So we have there are two clinical trials now in Germany, actually recruiting patients in a recurrent glioma, testing two of these inhibitors. One is a gap junction inhibitor, because these tumor microtubes of two different cancer cells meet, there's a gap junction. And this gap junction allows the flux of intracellular second messengers, and then calcium waves are conveyed over this network. That's how the network communicates with each other. And when you block the gap junction with the gap junction blocker MFA, that's the name, that's one of the drugs that is approved in the US for gastric ulcer, because it has also different anti-inflammatory activities, but it's also a potent gap junction blocker, and this is tested in one. And the second aspect is that on these long dendritic-like protrusions, there are these synapses between neurons and the cancer cells, and so we can block the synaptic input on these neural processes by giving an anti-epileptic drug that is exactly also blocking these malignant neuro to cancer synapses. The name is perampanel, and that's the second drug we are testing. That's a clinical trial I'm leading here in Germany, where we give it also to recurrent glioblastoma patients around surgery for recurrence, and we really hope that we can show also effects in patients. We have seen nice effects in animal models for both of these drugs before. It's also a nice anti-epileptic drug that I like to give to my patients with kind of treatment-resistant epilepsy in brain tumor situation. And so it's a certainly interesting drug to treat both, tumor-related epilepsy plus the neuronal input on the cancer cells. Yeah, there's a vicious cycle between these both. And the third drug is a drug that we are preparing to destroy at the moment. It's kind of in these networks, you have little pacemakers that, and this is similar to neurodevelopment. These pacemakers are generating slow calcium oscillations in the network, and they code in a frequency-specific manner distinct intracellular pro-tumor pathways in this cancer cell network. And there's a ion channel that makes the pacemaking, and we are currently preparing the trial with an inhibitor of these ion channels that has been in patients before, but not for any cancer indications. And now with all these insights from cancer neuroscience, we kind of get so many new ideas how to use drugs that are there, but also to develop new drugs that together with pharma to kind of target specific mechanisms of these neurocancer interaction.
Yeah. But I think what you're saying is that these clinical trials are ongoing. So is there a timeline that these will ultimately be concluded and you'll have some, specific data to work with? Or?
Oh yes, yeah. I mean, this all takes time. So clinical trials take longer than doing your in vitro or even in the mouse experiments, right? So yes, it takes. So I would say in about 2 to 3 years from now, we should have first data in. And it's the question is, what is the best strategy? What is the best drug? We will see. So we will learn from these trials, and hopefully we will then jointly in the field internationally develop new therapies. And not everything we are trying will work, right? That's otherwise, you wouldn't need clinical trials if you just think that you try works. But it's certainly great because everything else we tried so far in these specific cancers really did not work and failed. So we need new concepts, new biological concepts, and that's what we are now really aiming at.
Today's episode is brought to you by LightPath Technologies. From concept through prototyping, volume production, and global distribution, LightPath has the optical and infrared imaging knowledge and manufacturing expertise to be your optics and thermal imaging solutions partner every step of the way. Visit lightpath.com to learn more.
I definitely know that in the neuroscience field in general, I mean, you mentioned gliomas and the insidious way in which they spread and function, that traditional treatment has tended to fall short because even if there's a surgical intervention, there are, there's, I guess what you would say, a residue, some, leftover tumor that's in there that ends up, the person, that has a recurrence of cancer. And so that's something that clinicians have long struggled with. You kind of were touching on this already, but obviously these advancements have the potential to save lives. So that I always say with biophotonics, it's kind of a bridge between research and application. And how do you see the work that you're currently engaged in being put to use in the medical field, and what are your ongoing goals for the research?
Right. Yeah. So I think we really need to work on optimal concepts for clinical trials in terms of best patient population, best drugs, etc., which doses, readouts, trial concepts, and designs. So a lot of things need to be kind of worked on, clarified, and so we are collaborating on this. One thing that I like, and now reintroducing the biophotonics, so to say. So we are working here with our colleagues, neurosurgeon colleagues in Heidelberg, for example, on using intra-operative microscopy with new technologies to really study these communicating networks of tumor cells in the brain. And this could actually guide the neurosurgeons how much to resect, where to resect, and we can also learn in the live patient, so to say, during surgery, how neurocancer interactions and how, let's say, neural-like cancer interactions are really working. So we can learn about biology, but we might even be able to guide surgery by that. I mean, these are all dreams and ideas and hypotheses, but it's definitely important to underline that these during surgery, I think biophotonics is can be very a strong methodology that will help us to understand what is really going on in the patient, because that's in the end what we need, right? We need to be very sure that what we are doing is in the patient, and that the neural influences on cancer might guide then our differential therapies that we apply to patients.
As I understand it, your findings are laying the groundwork for cancer neuroscience, which is sort of a burgeoning field. So how do you see this field growing, and what collaborations do you see developing as a result of that?
Yeah, so the field is growing quickly, and there are many very interesting avenues. For example, how the brain itself in various brain regions is governing cancers in different parts of the body, right? Via the peripheral nervous system, that are the connections, or also by hormones shed in the blood. And there's an increasing body of evidence that there is really a very specific influence on brain activity in various different regions of the brain, in a very specific way, often on very specific parts of the body, and then also specific cancers growing in these specific parts. So it's kind of that is, I don't know, the next level of what we need to look into. Also kind of cognitive strategies maybe, or even this is something that you could consider. We know that stress, for example, is that we know for some time stress is not just bad for your well-being and bad for your quality of life as a cancer patient. It can even increase the growth of your cancer. So there's a lot of good reasons now. The same is true for pain, by the way. So stress, pain, etc., via neurocancer interactions can foster tumor growth. So there are many even more good reasons now why we need to reduce those in patients. So patient-oriented research will be very important to implement. I mentioned the neurosurgical projects. I think we need to talk also to psychiatrists, psychologists, etc. This is very exciting. Exciting is also the crosstalk between the nervous system, the immune system, and cancer. So the nervous system influences heavily how the immune system works. And the immune system, of course, is important now for all our great new anti-cancer immune therapies, right? Checkpoint inhibitors, etc., which work well in many cancers. However, many patients do not respond, or there are entities where it doesn't work at all. So there is now robust data that the nervous system is actually suppressing the immune system, hindering anti-cancer immunotherapies. And when you have released that and inhibit the neuronal input, then the immune system, T cells and other cell types suddenly are much more prone to attack the cancer cells and are killing the cancer. cells. And this is something that I find extremely interesting. And we are currently doing this in the brain for brain tumors. Also, this is kind of a new kind of avenue. Others have done it for extracranial cancers, where it's very clear that sensory and also sympathetic nerval influences are suppressing, by and large, anti-cancer immunity. So this is only shows that the complexity is always there in biological system and in diseases, and there are several more. The neurocancer connectome, so to say, throughout the body is something that we need really to map and understand. Influences on the various steps of the metastatic cascade. So if you suffer from cancer, the more than 90% of patients, of cancer patients, die of metastasis, right? So metastasis is a huge problem, and as it seems now with our combined data, we see that there might be really a sequence of neurocancer interactions governing this metastasis also. We certainly need to collaborate a lot, and Michelle and us, we have collaborated very intensively to kind of develop this neurocancer synaptic interaction story, and we continue to collaborate within the field. It's a very collaborative field, and this is actually, I mean, science is only successful when you're not sitting in your ivory tower or your little room, but if you kind of reach out, collaborate, and interact. And it's a fun field to be in. It's a great field. It's a very positive, collaborative field, cross-supportive field, and that's also something we definitely need to further develop. Yeah.
Right. Yeah, that's to me, that's what makes this work very exciting, is the interdisciplinary approach that you're talking about. I mean, because you're talking, on the one hand, you're talking about intrasurgical guidance to to generate a better outcome for the patient, but on the other side of it, you're talking about working with psychiatrists, and that's so I think holistic health is something that societally we've been working towards for a long time, and I think it's great that you're helping perhaps precipitate a change in that respect. But I do want to make sure that I take a minute to acknowledge essentially why we're here and why we're talking about this, and that is the Brain Prize itself from the Lundbeck Foundation. And do you see this recognition helping to foster research further?
Really? Yeah, the Brain Prize is an incredible honor, for Michelle, Monje, and me, but it's probably more really a great accelerator for the field. So it now shows, yes, cancer neuroscience is a kind of grown up now member of the neuroscience family. So we don't need to make the point that this is really neuroscience, and we need neuroscience. We need neuroscientists to understand cancer. So it will attract more neuroscientists to cancer research, more neuroscientists to cancer neuroscience, and this is what we need. And it will tremendously help to spread the word, to attract young, aspiring, or what's older young neuroscientists and older neuroscientists to dive into cancer, to use their background knowledge, their methodologies, all their wisdom, neuroscience wisdom, to really increase all the abilities that we have, and to really bring the field forward, push the field forward. And I am really looking forward to that. And I think the Lundbeck Foundation was very clear that it's kind of an investment into a burgeoning field. And as you said, and I find this a very so it's not so established like other fields of neuroscience where this is going on for 20, 30 years. It's more something for 10, 15, 20 years now that's going on. So it's a pleasure for us, but I think it's even more important for the field. And um, I think we'll try to make the best of it. It's also next to the honor, it's also a call for now collaborating even better. Developing the field now in the most vigorous way and bring the fruits to the patient at some point.
Mm. Yeah. Yeah, and I guess I'll just ask because again, at least in terms of the recognition, you and Dr. Monje are forever linked. And I know you've worked together, and I was interested to read related to the prize that you had had a conversation one day, and you started talking about something, and you realized that you had arrived at the same conclusion, coming at it from different angles. I imagine that you'll continue to collaborate with her going forward.
Yeah, well, we are kind of in a very good and close and friendly interactions, and try to coordinate things in a way that it's helpful for the entire field in the end, and that we are really have a focus on vigorous science, on making concepts and methods available, not just to each other, but also really to others. And it started actually. So here in this very room where I'm sitting, and we were sitting when she was visiting Heidelberg. We invited her for a talk, and she said, "Yeah, Frank, we discovered something crazy." And then I said, "Well, no, Michelle, we discovered something great." Then it took some minutes of interaction, and we then figured out that actually are there sinuses? There are sinuses on these, and so this was first. For a short moment, we said, "Okay, that's." You often think, "Okay, that's not great." If you have found something else has also found the same, but then because of our increasing good scientific and non-scientific also friendship, we decided we will build this together, and we will kind of develop the story together, and then we worked on it. We did a lot of interaction, and we submitted at the same point of time, and we resubmitted at the same point of time. This was very helpful, because if you report something that is quite wild, like synapses between neurons and cancer cells, then it's good to have another independent group reporting the same. And we found the same. So it was so robust. It was also good for us to understand that it's not some weird artifact of some models, but it's apparently a very robust biology that we are looking at. It's a hallmark of the disease. And that was also for us important, because we are fully motivated to study the functional consequences of this action. Because if it's so robust, if in biology something is really highly reproducible, then it's very likely that it's important and relevant for the disease. And so it was a great experience, and I'm really looking forward to continue this journey with Michelle and the rest of the field.
It sounds like, from our conversation to this point, the real obstacle is getting more people involved, right? Like intergenerationally, interdisciplinary. Is that the principal obstacle at this point?
Yeah. So
are there any like institutional issues associated with it?
No, I think the institutions are excited, and people are excited, and that's great. And Heidelberg and Stanford and Harvard and other places are excited about it and having this program. So that's great. Now, what we need to do, as you said, it's highly disciplinary. It's so we need to teach neuroscience to cancer people. We need to teach cancer to science people. We also need to now understand the peripheral nervous system better. So the central nervous system, the brain, spinal cord, this is quite well understood. We have we have a lot of more knowledge about the central nervous system than about the peripheral nervous system. And this is really understudied by and large in neurology, neuroscience. Only very few people in the world really focus on the peripheral nervous system. However, this is, of course, the point of contact to the cancer cells when we leave the brain, right? So we need kind of even basic anatomy, basic neurotransmitter and receptor classes are not 100% clear. The functional relevance, and then tools to interrogate it. This is all just in the beginning and in the making. And there are certain rabies and other dye tracing technologies that can be used to really understand which nerves and which neurons and nerves are really innervating these cancers and what is the functional consequence. And since the peripheral nervous system is spread out through the body, and often the nerve cell bodies are far away from the processes, this kind of makes it not so you cannot just apply typical single cell, let's say, analysis technologies, etc. So this really requires a lot of additional efforts and really new thinking. And for example, in Heidelberg, we have now together, Andreas Trump here and his team, they have injected dyes into the cancer. We have collaborated on them. It's just out, the paper. And so you could nicely trace the dye to the cell body of the neuron, which is far away. The spinal cord is somewhere. And then you can sequence these, and then you can understand, okay, what is the composition of these innervating neurons? And all this needs to be developed. It's not just there. Nobody has done it so far. We need to develop this in a novel way. And this is certainly a challenge, but also opportunity. And will also benefit neuroscience in the end, right? We will also kind of, it's a nice mutual beneficial interaction with cancer research and with neuroscience research.
This is exciting work that you're engaged in here to find new and innovative ways to explore not only the development of our understanding of brain cancer, but also the potential to intervene in therapeutic ways that haven't necessarily been tried before. And so I'm just excited that in the biophotonics community, we get a ringside seat in this process, because I'm very excited to see where your work goes from here. So I definitely want to thank you for spending your time talking with us today and explaining your work. And I wish you the best of luck.
Yeah, thank you so much for having me, and it was a great pleasure. And have a great day.